Are we ready to go, Rachel?
Yes, we're now live.
Good morning and a very warm welcome to this latest of our AHDB SCEPTREplus webinars. This morning's event is for tree fruit crops. And my name is Scott Raffle and I'm chairing the session this morning. I think many of you have been on these webinars before, but I just want to go through a few housekeeping issues. You can hear us, but we can't hear you, so you are muted. There will be an opportunity to ask questions this morning. I'll mention that in a minute. We aim to finish round about 11 o'clock or before then, so it's quite a short webinar this morning, and it will be recorded. So if you want to watch it again, or any of your colleagues or friends would like to watch it back, you will be able to see this webinar again. We have applied for basis points as well. So, because you are muted, you won't be able to ask questions without typing them into the question box. And there's a little mock-up here on the screen on the right-hand side. You will see halfway down that, if you look at my mouse here, there's a little grey tab for questions. You can click on the box there, type in your question and submit it, and we will try and answer your questions at the end of each of the presentations. Basis and erosive points have been applied for this event. Hopefully you will have been asked to fill out your basis details before or when you registered for the event. If you haven't done so, you can then send us your basis details at the end of today's event, and we will ensure that they go into the form and that we send off to basis. The event has been recorded and we hope to have that up and available, uploaded to our website by next week. So what are we here for? Well, AHDB has been funding a number of projects on weed control, apple canker and bacterial canker in cherry over the years. Most of the work we've done in recent times has looked at sort of, integrated pest management options for canker, and we ran a project number TF223, where we delivered a lot of information about that. But the one thing we haven't done is to actually look at alternative chemical products and alternative control agents for these problems. And so that's what the SCEPTREplus project is all about. It's a screening project. It allows us to screen alternative products to those that are being lost to you and any novel products that may be coming through the system. So this morning, we're going to look at 3 of these particular projects that we've been looking at tree fruit. And I'll just swiftly move on just to show you this morning's agenda. So we're going to kick off with Papp-Rupar at IBMR, who's been doing some screening work on Neonectria ditissima, which is apple canker. We will then have Chloe Whiteside from ADAS. She'll be talking about her screening trials on herbicides in apple. And then finally, we'll return to Papp-Rupar, who will talk to us about results that he's been getting for Pseudomonas syringae, or better known as bacterial canker of cherry. Now, we had hoped this morning to have Rob Saunders with us. Rob is our current tree fruit panel chairman at AHDB. Rob was going to try and help to put some context on all of this. We've had some technical problems this morning with Rob, so he may or may not be able to join us. He is there, but we just don't seem to be able to get hold of him at the moment, but we'll hopefully get him up soon. So I'm going to start off by inviting Papp-Rupar to share with us the results and the work that he's been doing on apple canker. Matt.
Well, good morning, everybody, and thanks for joining us for this short talk on how we're testing new products for control of apple canker. My name is Papp-Rupar. I'm from IBMR in Kent, and we're gonna first introduce apple canker for those lucky who have no prior knowledge of the canker. So it's caused by a fungal pathogen called Neonectria ditissima, and it infects through wounds. It's opportunistic pathogen. It infects through conidia, which are on your top right, the long spores, and the conidia are used for short distance splash and rain dispersal from branch to branch or between trees. And then it also spreads through ascospores, which are the spores in the lower picture of the 2 spore pictures, and they're produced by old cankers, and they are wind dispersed long distance, up to half a mile or more, so they can be the source of infections from old orchards to new orchards. Why I'm telling you this, because the canker, as you can see it here in the picture, like that left in the field, can produce both at the same time all year around. So in the middle you will have the ascospore production, the little red fruiting bodies, and the ascospores are reduced, while at the edge of the canker the conidia will be produced. So every canker left in your orchard can infect short distance around the tree where the canker is, and long distance. Now you can see from this picture and from your own experience, the Neonectria apple canker is causing cankers obviously on shoots and stems, and they're the result of the infection of lenticels, of leaf scars in the autumn, or picking or pruning wounds, and they can kill young trees and reduce yield and quality in mature trees, while at the same time Neonectria can cause post-harvest fruit rot as well, when it infects through the petal scars in spring. And probably the most important fact on this whole sheet is that majority of the commercial apple cultivars are highly susceptible and are normally riddled with canker in a few years. So how can we control it? First thing that we could do is remove infected branches and remove all the cankers and infected fruit from your orchards. You can do that in the winter, early spring. This will prevent your petal infection and fruit rot. You can prune it in the summer to prevent autumn infections, and you can remove fallen fruit in the autumn to prevent winter infections as well. Another thing that you can do, and our colleagues from New Zealand are huge proponents of this, is reducing foliar feed after harvest. All nitrogen put on leaves and trees will help, of course, the trees as well, but more importantly, in the canker sort of point of view, is that every bit of nutrient that Neonectria spores get at that time will make them much more, will make the infections much more severe, and the cankers much more severe next year. So they propose to reduce foliar feed to 0 after harvest. And apart from these two, removing, pruning out, and reducing foliar feeds, you could have used chemical control as well, though most of them are now in the past and not registered, and they were based on copper, and they were based on tabiconazole. And we used for applying during the petal fall to prevent post-harvest fruit rot, and during the leaf fall to prevent infection of picking wounds. Now, since none of these are registered for, let's say, picking and pruning wounds in the autumn, or leaf scars, we really need new products to control canker. And that's what we're here for. The strategy of this project was to select lots of different products and then put them through a lab screen first. We've selected products with the help of plant protection industry, SCEPTREplus panel helped as well, and we put together a wide variety of products that we think have a good potential. We have biological control agents, plant extracts, and new fungicides, all that are already registered or very close to registration. And we tested them in the lab for two main things. First, which is, to my belief, the most important one, is whether or not the products can reduce spore germination of the pathogen of Neonectria. If they can reduce spore germination, then there'll be no cankers. And if they don't reduce spore germination, then we tested the second thing that we tested was the reduction of mycelium growth, so reduction of fungal growth by the product, and that is whether even if it doesn't stop spore germinating, it can still help in the field by reducing the spread, and plant defense might be better, might be utilized better if the fungus is growing slower. So the aim was to select from 14 different products that entered lab screen, go down to the top 8 that would then be tested in the orchard trials. And we'll just go quickly through examples of the lab assays. So we tested mycelium growth. You can see the on agar plates on the top, where we put pathogen in the middle, and then the different products around it. And you can see in the far right as a control, which Neonectria is growing really well. And then the far left, the first two plates are two biocontrol agents that stopped or slowed down the growth of Neonectria, of the pathogen. While at the bottom, you can see three pictures of the spores. The one on the far left, you can see spores with no germ tubes coming out, so that product completely stopped germination. The middle one is water control, and the right one, you can see a product that really exacerbated the germination, so it provided nutrients instead of stopping the spores. So that can happen if when you apply foliar feed to the trees. The spores grow much faster, much quicker. So to the results, we split this in three different parts. So we have on the top biocontrol agents, on the middle we have new fungicides from the last couple of years, and on the bottom we have others such as plant extracts or plant defense stimulants. And we can see that if we look at biocontrol agents, we have three now entering the orchard trial because they've either inhibited spore germination or inhibited mycelium growth or both. We see that three new fungicides were inhibiting mycelium growth, but only one inhibited spore germination, and all three in green, you can see they went to orchard trials, while the rest that only have a partial fungicide, partial efficacy were not. The fungicides AHDB 9795 is a copper-based, and we decided not to put that in the orchard trial because it's extremely unlikely that this product will be registered. And then you can look at the other one of the other products. One of the have also very good spore germination inhibition and a little bit of mycelium growth inhibition. So we put that to orchard trial. So we have now altogether 8 products in the orchard trial. And what was quite interesting here, and that we used the previous standard, the previously used tebuconazole as a standard here, and we can see that actually it did not inhibit spore germination, only inhibited mycelium growth. So some of these products that we are testing now have the potential to be better than the previous standard. So to orchard trial, we have Braeburn mature trees, and we have very, very high canker pressure in this orchard. This is Braeburn Gala orchard, and all the Gala trees are affected, severely affected, and most of the Braeburn trees have a few cankers on them as well. We have a large trials with 18 trees per product in 6 blocks, and we have 8 products plus water plus tebuconazole standard. And we are applying products as you would in the autumn to prevent leaf scar infections. So we apply products between 10 and 90% leaf fall, and we gave the products the best chance of efficacy by applying the products first, and then we inoculated the trees, and then straight after inoculation, in 24 hours, we applied the products again, and then in whatever the interval was on the label of the product, we applied the rest of the doses. So hopefully we'll be able to see some results from there. So far we've applied all the products, we've done the inoculations, and now we're waiting to see the results from 3 different assays that we're running in parallel in the same orchard. So one is leaf scar protection with natural infection. So this is where we just expose leaf scars, and we count on the natural disease pressure, which is something that you would see in your own orchard. We have just to cover our backs if it would be a particularly bad year or good year for canker, then we put in the trial artificial inoculation as well. So that is simulating really high disease pressure when we put about 500 spores on every leaf scar. And again, here leaf scar was exposed, product applied, and then inoculated. And the third thing that we did, we exposed pruning wounds before spraying, and then sprayed them and inoculated them. And we're gonna just wait for a natural inoculation during the winter and fall in this case. So we're going to have 3 different data sets to cover 3 different disease pressures that can occur, or 3 different infection types that can occur in the orchard. We will measure canker incidence in April, and we normally do that quite late in the spring, because we want to make sure that all the juices are flowing, that the flowering is started. That's where you can really see the cankers being expressed, and you can be more or less sure that you see all the cankers that will be expressed. So we will be assessing and then reporting soon. So with that, I will ask you to stay tuned. We're going to have the final results by summer this year. And I'd like to acknowledge SCEPTREplus and AHDB for support, and also all the industry partners that were involved. Thanks to my colleagues that helped run the trial, and thank you for listening.
Thank you very much indeed, Matt. Very helpful. And yeah, I'm just trying to work out whether we've got Rob on the line. Rob, are you there? Rob Saunders?
Yeah, yes, I'm here.
Yeah.
Can you?
We've got Rob. Thank you, Rob. I'll just give you time to catch your breath. Couple of questions for Matt before I bring you in, Rob. Matt, AHDB funded project TF223, which was a 5-year IPM project, and we looked into a huge number of different possibilities of controlling apple canker in different ways. One thing that we did get out of that, which was really helpful, is we demonstrated that the use of Horticulture, which is tebuconazole, does actually work well as a wound protectant whilst pruning. So when that's actually applied through a chemical dispenser on the secateurs, either alone or in combination with the physical acting product blockade. Obviously, you haven't tried that in this trial so far. Is that something that might be worth looking at in future, and would that be any different from just a normal spray application?
Well, I haven't looked at it for one reason really, and that is most of the infections, or the one that are most difficult to control, are the leaf scar infection, because you have no control of when they happen. So they happen naturally. With pruning wound, you can sort of, let's say in best case scenario, choose dry days and dry days followed by more dry days, or prune where it's really cold, where the Neonectria infection levels are much lower. So you can do that, while in the leaf scar and picking wounds protection, you really have nothing. So that's where the sprays really come in handy. But yes, the Horticulture did really well in pruning wound protection. That's why I've included pruning wounds this year, to see at least at first glance if any of the products could be substituted if we completely lose Horticulture. Now I think it's only lost for post-harvest in apple, but you still can do it during the year, or blossom at blossom. But yeah, that's what we decided to do.
Okay, thank you for clarifying that. 2 or 3 other questions come in. One is comment about your water volume being very low compared with New Zealand recommendations. Was there any particular reason for that low volume of water?
Yeah, the trees are not that big. We checked, the spray cover was very good with 500 liters. And I had this discussion with Monica Walter, and they would say they have a slightly larger trees most of the time, and they would say the higher the volume, the better. But talking with with Adrian and other staff who do spray trials and testing different spray, they said at 500 liters per hectare with the trees that I had was more than sufficient. And we actually went down to 300 at 90% leaf fall, because we were just blowing through the tree pretty much, and not much was staying on. So we reduced it. And we still got, I think, a decent coverage. But yeah, okay. I agree. There's a differences there, and I couldn't really put my finger onto why New Zealanders go with such high. They go up to 1,500 liters per hectare sometimes.
All right, thank you for clearing that for us. Another question: why were 2 different Bacillus amyloliquefaciens used? Yes.
So they actually had a little bit different results, and the devil is in the detail. So the strains are very different. They belong to the same species, but what they produce in terms of bioactive compounds, in terms of how fast they grow in different environments, they are very different, or they can be very different. I don't know how different they are, but just based on my data on how they affect and how they look on the plate, or when you look at them under the microscope, and how they affect Neonectria, they are different. So strain is important with biocontrol. It's not all bacillus. For example, 99.99% of bacilluses won't do anything to Nectria, but these two are. That's why we have different strains.
Okay, so that looks quite promising. A couple more questions here, comments. Is the restriction on nutrition post-harvest only for nitrogen?
Well, I would say any foliar nutrient during infection, during high infection risk, which is autumn and spring. So you have low infection risks where the temperatures are near to freezing. There's very little sporulation there, or when it's very hot and dry. So then it wouldn't affect to have foliar feed that much. But when you apply foliar feed at the high risk of infection in autumn or spring, then you're basically adding nutrients for Neonectria to grow. And Neonectria is really well adapted to grow in very low nutrient for very long time. So other things might die off, but Neonectria won't. And when you add some nutrients, it's like pouring oil on the fire. I've seen the last results from New Zealand when they fed different amount to the roots, or put different amount of foliar fertilizer, and everything that you put on is increasing canker. So I would say some of the growers that I talk to quite often, they only use foliar fertilizer to spray the ground to increase leaf degradation, to remove inoculum for scab, but they don't spray trees anymore, and that's what they do in New Zealand as well.
Okay, one other question here before I bring Robin. What was the orange oil that you used?
Yeah, I would love to say. It's a commercial product. I'm not entirely sure whether I'm allowed to tell you what the.
No, well, you better not. You better not. The case.
Yeah.
But we can maybe pick that one up outside of the meeting if we need to. Can I just add at this point, because all of these SCEPTREplus projects are similar in that we have a lot of different coded products in them, and the reason we have coded products is that this is to comply with the requests of the agrochemical manufacturers. We have a good working relationship with them, but they do ask that products which are not currently registered or approved, or that they've not brought to the marketplace, are not actually named, and we have to respect that. So I know it's frustrating for growers out there who want to know the names of these products, but unfortunately, without doing it this way, we wouldn't get them at all. Rob, I'm going to bring you in at this point just for everybody's knowledge. I think most people know Rob Saunders. Rob Saunders works for Hutchinson's, and he also currently chairs the AHDB tree fruit panel, and we're very grateful for him joining us today. We've got hold of him. Rob, just wanted to put some of this in context. First of all, comments come through to me that one grower says he can't get supplies of Horticulture this year. Can you update us on that? What's your understanding?
Okay, there are several brands of tebuconazole, and they're all basically falling by the wayside in terms of their registration, or their label has become so restrictive that they contribute nearly nothing. So yeah, not surprised to hear that you can't get hold of them, but this is one of the reasons why the work that Matt is doing is so important and so relevant to us. I've got actually one more question, if I can put to you, please, Matt. And that is that looking at your results, I see the orange oil was good at inhibiting spore germination, and the potassium phosphate was good at inhibiting the mycelial germination. Can I invite you to speculate what might be achieved if you applied those two things in mixture during leaf fall?
Yeah, I would love to do that, but I mean, that's the next stage, is try to figure out which products can be mixed and at what ratios. But I think that's a really good way forward, to have a basic orange oil that acts as almost a barrier that protects spores from germinating, and then if that gets washed off, maybe not together, maybe one after another might be a better option, but I can't really comment which one would be better now before we test. But that's definitely something that they can, or even we don't know if these, or if orange oil or phosphonate have any effect on bacteria, on bacillus. So in best case scenario, they don't, and then you would be able to use bacillus throughout your harvest already. So build a population of bacillus on the tree, and then during the leaf fall, add orange oil and phosphonate, and that it gives you very sustainable protection. I'm not sure how expensive would that be, but it would definitely be very, very, very nature friendly or environmentally friendly protection to compare to anything else.
Brilliant, thank you for that. So you've obviously got really good communication relations with Monica in New Zealand, and she's really strong on the cultural controls, and she talks about just pruning out cankers on a regular basis as being one of the most powerful techniques. Has she given any indication of products which New Zealanders have access to and are making making progress with?
Yeah, so they have a very mixed, very mixed results. They've tested some of the biocontrols that are similar to what we're using. So they've already tested 1 or 2 bacilluses, and they haven't found anything. But I've talked to her about the way they're testing, and I think their assay it favors fungus way too much, so they put way too many spores, and they make everything absolutely optimal for pathogen to infect. So that's where it's very difficult to see. That's why we're doing 3 things at once to give it a range of infection pressures. So they don't see very much effect in their climate and with their methods on bacilluses and other biocontrols, but we're still hopeful that we will see that here. And what comes for the chemicals, they still have tabuconazole, no problem, and they even have Captain for post-harvest as well as tabuconazole. But I'll just like to point out what you said. It's absolutely true. If you ask her, they've done a trial where they've done a 5-year trial where they doubled either amount of sprays they put on or amount of pruning that was done. And the sprays doubling or even tripling the amount of sprays did little to decrease canker without actually stepping up in pruning. So when they stepped up pruning by 50% or more, so going in more often or cutting down more severely, they actually in 5 years brought canker incidence from 40% to below 10 on the 2 or 3 trial sites. But that is really expensive to do. So that's the problem.
Okay. I'm conscious of time. We've got 2 more talks to give. So, Rob, if that's all right with you, I'm just going to draw this to a close. But just before I do so, there have been a few more comments coming in. As regards the different strains, Alex Radu from Agrovista was just making the point that different bacillus strains will have different fungicidal effects. And then a couple of comments on tebuconazole. Jonathan Blackman at Hutchinson's is making the point that the manufacturers have stopped making the 200 gram per litre products of tebuconazole, which are the ones that do have authorizations. He's making the point that the actual product Folliker has not been available for some years. And again, Alex Radu from Agrovista is making the point that the product Deacon, the map number 19003, can be supplied until the 28th of February this year and allows application to top fruit for this autumn. So thank you for your comments, everyone. Thank you, Rob, for your helpful information. And one last comment that's just come in is somebody's making the point that in New Zealand they are moving towards cutting out every month. So Thank you. We've got to bring that to a close. I think this interesting. We've got so many comments here. The important thing to say at this stage is that the final results will be in shortly, and we will, of course, make those available to all the industry just as soon as we can. So, Matt, thank you for bringing us up to date with your project on apple canker. We will move on to weed control in apples now. And for this purpose, we've got Chloe Whiteside from ADAS. This particular project, looking at alternative herbicides to the existing products that we've got, was undertaken by Sonia Newman of ADAS, but unfortunately Sonia has moved on to pastures new. So Chloe is stepping in and is going to explain to us what Sonia and her other ADAS colleagues found in this particular project. Chloe, the floor is yours.
Thank you, Scott. Thank you for that introduction. Can I just confirm that you can also see my slides?
We can.
Excellent. Right, good morning, everybody. Thank you for tuning in. So yes, this morning I'm going to be talking through some work that we carried out at ADAS on behalf of AHDB through the SCEPTREplus project, looking at weed control in apples. So just to give you a bit of background as to why we were doing this work and why it's important to look at. So within the UK, the tree fruit plantations, they cover approximately 24,500 hectares, and a large percentage of this area is apples and pears. And in terms of value, well, dessert apples alone, they're worth around 105 million a year to the UK economy. But unfortunately, tree fruit crops, they are weak competitors against weeds, particularly at the early stages, so when they're young trees, a couple of years old. And weed infestation can result in yield penalties of 10 to 20%. So this, it equates to a big loss, a loss of at least 10 million per year for growers. So it's really important that we keep trying to look for new actives and ways of controlling our weeds, because they can become really problematic, and it's not always easy to achieve good control. Now, there is gaps in both the contact and residual herbicides for young tree fruit plantations. So within SCEPTREplus, we did carry out 2 trials last year looking at a range of contact products and residual products on 2 different sites, just so we could try and find products that are both effective but also crop safe on young trees. So I'm going to talk you through the contact trial first and just give a little bit of background to what we did, and then more importantly what we found out. So this was carried out at a site in Kent, and we were looking at the Brabham variety. In terms of the application, so we just put on 1 herbicide application after the trees had broken bud, and this was using a single nozzle hooded lance. We always have an untreated control in there to compare against as one of the treatments. We also had the grower control product, so this was a glyphosate product, so then obviously we can compare the efficacy of these new products against what we're already using as a standard. And then we had 4 experimental products in there. So you can see in the treatment table that I have that they are all coded, and I apologise for that. As Scott said, we have to keep new things coded until, if they come through to market, then obviously we can release the names of those products. So I appreciate that there's some frustrations around that, but that's just the way we have to have it at the moment. So 3 of those experimental products, they were tested in a separate SCEPTREplus project on bush fruit in 2018 to 19. So they're things that have been around and we have tested previously, and they showed promise, so it was good to try and then look at these on tree fruit as well. So as I said, 1 application went on in May, and then we had a look for efficacy and any signs of phytotoxicity at 2, 4 and 10 weeks after that treatment went on. So what did we find? Well, in terms of crop damage, we didn't see any phytotoxic effects from any of those 4 experimental treatments. So in terms of that trial, all of those treatments were crop safe, and just as good as the grower standard in terms of the safety on the crop. Looking at the efficacy, now 2020 was an unusual year for many reasons, and one of those things being that it was extremely dry from around April to June, particularly in Kent. So naturally weed cover was low throughout the trial anyway. So that does have to kind of be taken into consideration when you look at the efficacy of these products. The trees were irrigated, so we had trickle irrigation, but this didn't really appear to affect the weed growth, not within the trial, not within the commercial crop either. But as time went on, it didn't stay really hot and dry forever. So we did start to see an increase in the weed cover across all the plots at the final assessment in July. So this was 10 weeks after the herbicide application. But bearing in mind as well, you know, we're looking at contact products here. So you would expect that after 10 weeks, because we haven't put residuals on, you're going to start to see new weeds coming through anyway. The main weed species that we were getting within the trial area were creeping thistle, knotgrass and annual meadowgrass. So if you have a look at the graph that I have here, really we want to be looking at the blue bars. So that was like the final assessment on the 22nd of July. So we have the untreated down the bottom here, and then the products going up here. So you can see that, you know, this is the grower standard, this was doing very well. And then here we've got the experimental products. But as I said, remember that we're kind of starting to see weeds coming through at that stage anyway, because we were just looking at the contacts. If you look at the bars, the different coloured bars earlier on in the trial, so from the 27th of May and the 9th of June, then actually everything is all very low and very comparable with the untreated. So not really much to pick out there in terms of those products earlier on. There's nothing kind of jumping ahead or falling behind in terms of its efficacy earlier on in the trial. So I'll come back to kind of how we take that forward in a moment. In terms of the residual trial, so this was carried out at a site in Gloucestershire, and we were looking at the variety Gala. And again, it was carried out in a similar way to the contact one. So we just had the one application, which was done at bud break, and we had a single nozzle hooded lance. Similar kind of layout with the treatments. So we had 4 experimental products, some of which had already been tested in previous trials on bush fruit. And then the Stomp, Apca and Flexidor was the grower control that we were comparing against here. So this went on in April, so slightly earlier than the contact trial. And so we did have some rainfall within that month. So, you know, it wasn't too hot and dry. So we had a good amount of moisture to really get those residuals going and get that active going in the ground. We looked at assessments 2, 4, 7, and 12 weeks after the treatment, and we also looked later on at harvest at shoot extension and tree girth, just to see if there was any knock-on effects to that from any of these residual treatments. So what did we see here? Well, again, in terms of the crop damage, we didn't see any phytotoxic effects from any of the 4 experimental treatments. So as far as that trial was concerned, they were all crop safe, and we also didn't see any effect on the tree girth or the shoot extension when assessed at harvest in September. So from a crop safety point of view, those products were really promising. The weed cover was initially low because, again, as I said, it wasn't the ideal conditions for weeds, so it was very, very dry as time went on into the trial. So the weed cover was initially low, but after 12 weeks, we were seeing that the cover of those weeds has actually gone up to almost 59% of total plot cover. So it really had increased. And we did get significant differences at the end of this trial with all the coded products reducing the weed cover. So looking at this graph, again, that really is starting to stand out. If you're looking at this dark blue line here with the untreated, where we're almost up to 60% at the end of the trial, and then looking down at these experimental products where we've got around 25% and less. So really giving us some good efficacy data there. So we saw the lowest levels, which were in the grower standard, Stomp, Apca and the Flexidor, but then that was quite closely followed by this experimental product here, 9947. And we were seeing a range of species within this trial area, mainly groundsel, field bindweed, fat hen and redshank. So these products are giving us good efficacy and good control of a range of different weed species. So in terms of some take-home messages and what we can kind of learn from that, from the contact trial, to reiterate, they all seem to be crop safe in this trial and no phytotoxic effects were seen, but you have to bear in mind that it was extremely dry from April to June, so this did kind of have an effect on the weed germination. So really it would be better to have another look, if we could, at these products under less atypical conditions. So, you know, can't always base your results just on that 1 year. It's good to have a look at another year when the conditions are maybe slightly different, just to kind of look at the efficacy of those tested products under different years. Now, as I said, the products, they are all coded. We can't release the names to you, which I appreciate is frustrating, but there are a couple of things that are already out there on the market on different horticultural crops. So 9868 and 9859, we already use these under emus for different horticultural crops. 9868 has good efficacy against black nightshade, annual meadowgrass and other broadleaf weeds. So in terms of if the crop safety allows and the emus can be progressed, then having things that are already authorised under other emus does make that process a little bit quicker. 9897 and 9921, not currently authorised for use in horticultural crops, so getting those onto market could take a little bit longer, but that kind of gives you some hope that there are some promising products out there, and hopefully we can try and get these through to market. In terms of the residual trial, just to reiterate again, all seemed to be crop safe and no phytotoxic effects were seen, and it didn't appear that the residual herbicides had had any effect on the growth and the vigour of the trees when they were assessed at harvest. But as I said, with the contact one, it was generally dry throughout that trial period, and so it would be good to, if we could, have a look at the products again in different years and kind of different weather conditions. All of these coded products did reduce the weed cover significantly, so really promising in terms of their efficacy, and particularly 9947. So that's a product that again is already currently authorised for use in other horticultural crops, mainly in outdoor ornamentals under an EAMU. So there is the hope that if this product is, you know, promising and can be brought forwards into use on tree fruit, the fact that EMU is already there could hopefully improve the speed of that process. 9918, that's also got an EAMU for outdoor ornamentals, and that product in particular is very good for use on grass control. So hopefully that's one that we would be able to bring forwards in the future. The other two, 9898 and 9900, not currently authorised for use in hortic crops, so a little bit further away in bringing those ones onto market. But the good thing here is that there are some products, both contact and residual, that are showing promise. They've got good efficacy. They've been shown to be crop safe within this trial, and hopefully if we can bring those forwards, then that wouldn't be too far into the future that we could do that. So that kind of brings me to the end really, and just some acknowledgements. Again, AHDB for funding the work and the chemical companies that supply the products. I'd like to thank the scientific staff that actually ran a lot of the work for us at ADAS Boxworth and Rosemond, and obviously thank you to the growers for hosting the sites and for their technical input into the trial as well. So Scott, that brings me to the end, and if there's any questions.
Thank you so much, Chloe. Yeah, just to reiterate Chloe's point there, my colleague Adam Doxford and our crop protection team really it makes his life so much easier if some of these products have got approvals on other crops. So the fact that two of the contacts have got emus on bush fruit and asparagus rhubarb is a great help, and similarly on the residuals with their approvals on outdoor ornamentals. So we will be working with Adam on these to see if we can take those further forward. Rob, can I bring you in at this point? You know, how difficult is it for tree fruit growers controlling weeds at the moment, and how urgent is it that we get these alternatives?
There's been a tension in the last few years, hasn't there, between what's required from a residual weed killer particularly is something that something that is persistent for a period of time, and also something that is just slightly water soluble, so that it can be taken up by the weeds. But if you're working for the Environment Agency and the water boards, what they want is something which breaks down very quickly and isn't at all water soluble, so it doesn't turn up in the water supplies. And those two tensions in the last few years have meant that we just haven't seen any fresh residual weed killers come to market and gain approval for horticrops. So this has been such an important piece of work, so relevant for the industry. I know growers have struggled to do a good job. So Chloe, I think this has been a great bit of work, and as ever, slightly frustrating that it's all presented as coded products, so that it feels like jam tomorrow, but it's a great piece of work.
Okay, thank you, Rob. My colleague Rachel McGauley, who's our research manager for tree fruit, is listening into all of this, and I'm sure she's scribbling lots of notes. I think the one thing we would have to consider is whether we want to do any further work on this, Chloe, and to repeat this, because I do appreciate 2020 maybe wasn't the ideal year for doing weed control trials, given the nature of the spring and the dry weather we had. Just to finish off on this topic, I'd just like to say my colleague Adam Doxford is currently exploring two other options. One is wing P, which is dimethenamide and pendimethalin mix. That I don't think there's any prospect of getting that in the near future, but we are talking to manufacturers and exploring whether we could possibly get an EMU for that. Slightly further down the line is the contact acting herbicide Shark, which I understand I think is awaiting re-registration, but we have undertaken some residues trials on that. We've gathered some residues data which will support uses on all tree fruit crops. So watch this space. We are looking at Shark as a possibility. We must move on. One other sort of one last comment that's come in, and I don't want to ignore those kind people that are sending comments in, that there are certainly two possible contacts for further study here. So it certainly has not been a waste of time. This has been good information that we've gathered. And Chloe, thank you to you. And if you speak to Sonia, thank you to Sonia as well for all your work on this. Thanks for now. Yeah. Which takes us on to our final presentation of the morning. And we're going to bring Matt Papp-Rupar back in again. We're moving off apples onto cherry this time, and Matt is a pathologist at NIABMR who works on a lot of disease control projects. One of these is bacterial canker in cherry. You've possibly heard him talking about the other projects that we've been doing on this. Matt's going to talk us through the latest work that on SCEPTREplus project are looking for alternatives to copper. Matt.
Right, I hope you can see my presentation, because I have no idea whether
Yes, we can. It's fine.
Ah, perfect. All right. Okay, so I'm back for the last talk, and we're going to go straight into another canker, cherry canker this time. And as with previous one, I'm just going to recap a little bit of disease cycle and what we're trying to prevent. So it's caused by, as a title suggests, by bacteria, Pseudomonas syringae, this black and white little rod-shaped bacteria here in the picture. In UK, we have three strain groups, Pseudomonas syringae, and two more Sprenorans races that are the most important pathogens. And the disease cycle, you can see pictured here, is quite complex for a little bacteria, which is you either get tree pre-infected somewhere and you have a canker already on it, or bacteria can be brought in by wind and rain into the orchard in the spring, where they live on surfaces not causing anything, so you don't know that they're there. In the summer, on some cultivars, not all, if the conditions are right, the population will increase on the leaves, and some will go in the leaves and cause these shot hole symptoms, and also cause some necrotic spots on very young fruit. So that's early summer. And then population keeps increasing on the leaves throughout summer and autumn. And then throughout the leaf fall, this population of bacteria from the leaves is washed into the either in the buds, leaf scars, and other wounds. And then more wounds are created, sometimes in the winter, with frost damage And Pseudomonas has one really neat trick, and that is can induce frost damage by making ice itself to make openings in the wood to colonize it, and then you get cankers in the spring, which are huge sources of bacteria then to recycle in. So what we're trying to do is cut the cycle in late spring, summer, and autumn. Just cut it straight down. So reduce population in summer, which will then reduce population in autumn, reduce chance of infection, which will then reduce canker incidence the next year. And so far, this was done mainly by copper-based products. And now this project is looking into some very short-term replacement, direct replacement for copper. And we're looking at bactericidal chemicals, biocontrols, and then some immunity boosters and tree health boosters as well. So these are the products that we're testing. We have two controls, inoculated and non-inoculated, and then we had one of the products that was previously used, just as a comparison, how well these products are working against the previous one. And then we had some plant extras. We have some bacillus, and here you actually have two products that are not coded because you can use already on cherry, I believe. And then you have some completely new products with some elicitors, some bactericides that we're testing. We are doing this in a polytunnel on a very susceptible sweetheart cultivar, and we have 12 trees per treatment in 4 blocks, and we have a mixed infection of 3 different bacterial strains to sort of mimic the natural conditions. And we applied products with the knapsack sprayer with 500 liters per hectare for these small trees, which is it's quite good coverage. And then we inspect phytotoxicity after several treatments. What we did then after phytotoxicity was inspected, we counted how much bacteria is surviving on the leaves in different points in the season. And then the following season, we measured symptom prevalence and symptom severity on these trees. So just quick application and timing. Some of the biocontrol agents were sprayed up to 7 times from beginning of August till the beginning of October in about 10 to 14 day intervals. But some other, for example, like copper, were only sprayed 4 times at this dose to give you the maximum dose that you're allowed in a single season. And we've enumerated twice the bacterial concentrations on the trees in the beginning of August, and then again in the end of September, to see how these products are doing at the beginning and then at the end again. So starting our results in August, these products were applied at this point only once, and they were applied prior to inoculation. So they were already on the trees, then the trees were inoculated, and then 2 days later we looked how much bacteria survived. The red line is the amount of bacteria that we put on if you measure them straight away after the leaves are dry. And you can see that in all products, even when you have just treated with water, in 2 days, about 90% of bacteria die already. Mind the logarithmic scale here. So 4 is 10,000 bacteria per square centimeter, let's say, and 3 is 1,000 and 2 is 100. So the difference between this product here and the water is about 10 times. The bacterial concentration here is 10 times lower, which means that this product has reduced population just with one preventative spray by about 90%, while these two, which have a star under signifying that the difference versus control was significant, these reduced, they don't look very different just you look at the graph, but they actually reduced by about 70% the population size just by one preventative spray. Now we sprayed, as you saw on the previous slide, throughout the August, throughout the September, and then the end of September we measured again how much bacteria is now there. And the first striking thing you can see here, that when I was talking about the disease cycle, you can see throughout August and September everything got much closer to the red line now. So bacteria population in all treatments increased by about 10-fold in this 10 weeks or less. actually 6 weeks. But some products, for example, this product, the AHDB 9829, was very good at the single, reduced at the single treatment, is still very good. But then the Serenade didn't do anything after the single treatment. As expected, as a biocontrol, it needs time. It was actually as good in September as this fungicide here. So this is quite interesting. Result means that you can achieve some control, and this is again about 90% reduction in bacteria population. So if, let's say, if in water we have 1,000 bacteria per square centimeters, these only have 100. And untreated, not inoculated, which are the lowest, are not that much lower. So even if you don't inoculate, you still get quite a lot of bacteria growing naturally there. So these were actually quite good. So these are at the end of the season, just at the beginning of leaf fall, where you would expect that these populations that are present now are the ones causing you trouble throughout the winter and the next season. So we checked that, what happened in the next season, and we did two things. One is we assessed how many of the leaf buds have survived and made nice leaves next year. And we considered these, you can see here 2 different pictures. One that only has 2 little arrows, the 2 leaf buds. This is one very healthy shoot. Just 2 leaf buds didn't develop. And here about 80%, 90% of the leaf buds just shriveled after they've opened. And these were all infected. And you can see this is quite interesting. The 2 products that were very good, so this product AHDB 9829 was very good at the single, after the multiple applications, and again reduced symptoms incidence, reduced these symptoms by about 60% versus water control. And there were 3 other products, they also reduced by about 30% incidence of this bud dying off. And I have to say, this was assessed in very, very high disease pressure. So untreated, non-inoculated, this is your normal disease pressure. If you don't spray anything, but you don't add extra bacteria. So about 7 to 8% of leaf buds may get this symptom anyway. And we had much higher, especially in water. And 2 other products didn't really do much. But then when we were assessing this, we realized that some trees have more or less of these severe symptoms. These symptoms that are actually really big sources for your infection if you don't remove them throughout the season. And they are these bleeding cankers either between the buds or straight out of the buds. And again, there's only one product that was now significantly reducing versus water control. But interestingly, there were products who made it worse, not significantly worse, but worse nonetheless. And these, the Serenade and the Milo-X, the BCAs, and the other product that were affecting at the end of the September, they have reduced now, not significantly, but they have reduced the severity. So the trees had less of these severe canker lesions. And again, I have to point out, this was tested in very high disease pressure. So to summarize it, we have this fungicide that it had been sprayed 4 times altogether, and it was really good at single dose, at multiple dose, and then it decreased incidence and severity of the disease. You have the BCA, the Serenade, which as a single wasn't very effective, reduced a little, but as a multiple it was quite effective. It reduced incidence significantly by about 30%, and the same with severity, but maybe less significant. You have one defense elicitor that didn't do much for the pathogen population, but it did decrease reduction. It reduced, disease incidence, so there was less buds that died off, which could be used together with the BCA. And then you have the other biocontrol, the Milo-X, that did pretty much the same as the elicitor, and the rest weren't very successful, apart from this one here, which was very good at single spray, but unfortunately this is a completely new product, and we had the dose wrong, so it was phytotoxic, and we weren't able to, the trees were damaged. So I think that then when you damage the tree, you make openings for the bacteria to come in, so it wasn't effective there on. But if we work on improving the dose here, I think this product has a really good, can have a very good future because it's a food grade product. So it should be very easy to register. So in conclusions, Serenade with a 14-day spray interval from August to September was good in reducing population and symptoms the following year. The two other products, the Elicitor and the Milo X, had a little bit of control of either bacterial population and a little bit of control of incidence, but not severity. The copper-based AHDB 9829 was the most effective and could offer direct replacement for the previous, but it's not very likely that it's going to get approval. We might, if now we show that it's very effective, especially for high-risk young orchards, that the whole trees could be killed. It might grant EMU there. And then the bacteria side, as I said, showed some encouraging results, and we really need to fine-tune the dose to avoid the phytotoxicity. And with that, again, acknowledgement to the funders and all the colleagues who helped me on this project. So I'll open to questions.
Thank you so much, Matt. Well, I would say there's some bad news and there's good news. Let me start with the bad news. The copper-based product, AHDB 9829 that you mentioned, which is obviously giving the best results there. The bad news is my colleague, Adam Doxford, who works closely with CRD, he's had no success at all in trying to secure emergency authorizations for any copper-based products, either for canker or for scab and apple for organic growers. And we just don't see a way forward for that. There are issues in terms of the environment, which CRD and Health and Safety Executive are not happy about. So much as I'd like to say that's good news, it's highly, highly unlikely that we'll make any progress. Good news, I think Serenade obviously shows some potential. And I just wanted to draw in one of the results from our IPM project, TF223, which looked at bacterial canker as well. And the cultural trials in that project demonstrated that extending the period of rain protection from prior to blossom until leaf fall actually led to the largest reduction in canker spread in orchards which had existing infections. So I'm wondering, and I'm going to bring Robin here, I'm wondering whether a combination of that cultural control technique, growers extending the period of cover, coupled with maybe Serenade ASO, is that something worth trying? What do you think, Rob?
Yeah, definitely. I've always been slightly puzzled as to how we, the task that we're trying to do here in effectively driving down bacterial numbers on leaves is extraordinarily difficult when you consider that the medical profession can't sterilise an operating theatre. So, you know, how are we to reduce bacterial loading on plant tissues? But so this is it's a significant task, and clearly it's going to be a combination of cultural and spray applied approaches, which is likely to give us the best effect.
Yeah.
Good. Agreed. Yeah. Any other comments, Matt, on that?
Yeah, I mean, the comment and maybe the problem with the trial that I have is that my trial was in polytunnel the whole time. I mean, we put the covers off after the leaf fall, after we've done the trial, but that is protected like our TF223 results suggested, that keep the covers on. I mean, I have used that to keep the conditions as constant between the different sites, and I have inoculated to accommodate for high inoculum to have a disease pressure. But as a rule of thumb, the more wet days you have, the days where the leaves have moisture on, the more bacteria will grow there. Bacteria can boom, multiplied by hundreds and hundreds, factored by 100 in 2 to 3 days if it's warm and rainy. So having a cover on even for a couple more days, if you expect the rain but not wind, will definitely decrease bacterial populations.
Yep. Okay. Well, there's a number of things that we can take up with Adam again as a result of this project. Conscious of time, Matt, thank you so much for your time this morning.
No problem.
I'm going to bring in my other presentation that we were looking at earlier on. We'll move over to mine, and we'll just bring this in, hopefully. I'm just trying to move that one on. Here we go. So first of all, I'd just like to thank the speakers this morning. I'd thank Matt and Chloe for doing a splendid job explaining so clearly the results of these projects. Thank you to Rob. I think the purpose of bringing Rob in as our tree fruit panel chair was just to try to put a bit of context into things. It's very frustrating doing webinars when you can't bring members of the audience in to discuss issues. So I'm grateful to Rob for giving us his time this morning to do that. I'd also just remind you about BASIS and Neuroso forms. As I said earlier, there was an opportunity for you to complete those at registration stage. If you haven't done so, but you still want to submit your make an application, submit your details to us, and we will add you to our form that we submit to BASIS and Neuroso. You can send it either to me, scott.raffle@ahdb.org.uk, or Rachel, our organizer for this morning, rachel.morrison@ahdb.org.uk. If you do have any further questions about any of the topics that we've covered this morning, again, send them directly to me and I'll pass them on to the presenters and we'll get them to make contact directly with you. Just as a reminder, as I said at the start, this event today will or has been recorded and it will be made available next week on our website. And just to finish off, there will be another webinar, an extended webinar on the 25th of February. Many of you normally come on an annual basis to our AHDB Tree Fruit Technical Day, which normally takes place at the Orchards Event Centre at NIABMR East Malling. Obviously, because of the pandemic, we won't be doing that in its usual form this year, and we will be doing a webinar. We have decided to run the webinar from 1 o'clock lunchtime through to 5:40. A number of growers have requested this in the past, saying buds are moving in late February time and they'd rather be out and about doing things in the morning. So we've specifically timed this to start at lunchtime to run through the afternoon. So hopefully you'll get more time to join us. The purpose of that Tree Fruit Technical Day is to cover results from all our other AHDB and other funded research into tree fruit. So we'll be covering such things as TF223 project, which we've talked about a lot today. I'll be giving a brief summary of the results that have come out of that 5-year project on IPM in tree fruit. We'll also talk about the Plum Demonstration Centre and the results that have come from that this year. And we'll talk also about a number of other crop protection issues, dealing with apple canker again. We'll also deal with woolly apple aphid, some soil amendments issues that we can follow for tree health purposes. And of course, don't forget spotted wing Drosophila on cherries and plums. So we'll be covering that comprehensively. So if you want to join us for that, if you search for the event on the AHDB website, if you just search for AHDB Tree Fruit Technical Day, and you'll find that on our events page, you can register for that to join us on the 25th of February. So I hope you've got something out of this morning. I hope you've benefited from the results, and we do hope to hopefully see you on the 25th of February. But in the meantime, very good morning to you all. Thank you.
Thank you, Scott.